Serotonin is an evolutionarily ancient neurotransmitter system, important for basic behaviors such as feeding, sleeping, eating, and even in the regulation of emotion. Hence, it comes as no surprise that dysfunction in serotonergic neurotransmission have been associated with neuropsychiatric disorders, most notably major depression. In fact, the most prescribed class of antidepressants today are SSRIs, or selective serotonin reuptake inhibitors, which are thought to act by increasing concentrations of serotonin in the brain. The brain is a heterogeneous organelle comprising of several different cell types. Even amongst neurons, there are several subtypes of neurons, for example, glutamatergic, GABAergic, and serotonergic. In order to study neurological disorders and even model them in vitro, it is becoming increasingly clear that we must first generate the relevant neural subtype in vitro. For example, in the case of neuropsychiatric disorders, we must first generate serotonergic cells in vitro in order to study serotonergic neurotransmission in a context of disease. One way to generate serotonergic neurons in vitro is to utilize pluripotent stem cells. Dr. Yamanaka's Nobel-winning discovery shows that by overexpressing a key set of transcription factors, one can reprogram human skin cells into a pluripotent stem cell stage. These are called human pluripotent stem cells, or induced human pluripotent stem cells, or IPS. These IPS can then be made to differentiate into any other cell type using the right combination of growth factors, small molecules, and developmental signaling molecules. An extension of this concept is to overexpress key transcription factors that can directly convert skin cells into a target cell type, such as neurons. And this is called direct conversion, or transdifferentiation. Utilizing IPS as well as transdifferentiation technologies, within the last years, 3 groups, including our own, have published methods to efficiently generate human serotonergic neurons in vitro from human fibroblasts, or skin cells. 2 of these studies utilize transdifferentiation, or direct conversion technologies, overexpressing key transcription factors that promote serotonergic transdifferentiation in vitro. And the 3rd most recent study utilizes a combination of growth factors and developmental signaling molecules to differentiate serotonergic neurons efficiently from human IPS. Each study generates 25 to 50% serotonergic neurons in vitro from human fibroblasts or human pluripotent stem cells. The generated serotonergic neurons are all electrically active. They release serotonin in the culture media, express certain markers of the raphe in the human brain, and they are capable of releasing serotonin in a way that is modulatable by SSRIs, suggesting that this may be a novel platform via which we can model neuropsychiatric disorders using patient-derived serotonergic cells. Each method has its own caveats as well as advantages, and this is covered and reviewed in our recent BioEssays review.